I'm doing right now, because he was the person convinced Randy at the time that we should study regulatory T cells when everybody is mostly focusing on B cell and CD40 signaling at the time. So he really started all the regulatory T cell work in Randy's lab. And after he graduated, he moved to June Allison's laboratory at Memorial Sloan Kettering in New York to continue study regulatory T cells and immune regulation, but in the context of immuno-oncology. So there, he studied how CTLA-4 blockade works. So we know CTLA-4 is one of the top immune checkpoint regulator, along with PD-1, that everybody studied these days. But early days, that people always focusing on was always focusing on the effect of CTLA-4 blockade on the effector T cells, because we know CTLA-4 inhibit T cell activation. But we also know regulatory T cell express high level of CTLA-4. So when he was in June Allen's lab, he showed that the CTLA-4 blockade actually act on both effector T cells as well as regulatory T cells. And when he started his own laboratory, his own group in UK at UCL, he really showed how the CTLA-4 blockade, CTLA-4 antibody can work on Treg, and that work really opened up a new direction in terms of studying how using an antibody approach to deplete Treg cells, and also the depletion of regulatory T cells is depending on the Fc receptor, the Fc portion of the antibody that you use. And really, because when we study antibody, most people really focus on the antigen it recognizes. And even though when we were in college or when we first learn immunology, you always have to learn Fc receptor, but this is a part that people really don't pay enough attention to. We usually forget, and we just have to remember because we have to take the test. But there's different type of Fc receptor, different cell express different type of Fc receptor, and those Fc receptor really are the key molecule that dictate the biological activity of the antibody. So by really going into this direction to know what type of Fc receptor that can better, make this antibody work, he really opened up a new direction, not just study anti-CTLA-4, but recently to study the anti-IL-2 receptor, anti-CD25, as an approach to deplete tumor-infiltrating Treg cells, and also by modifying the Fc receptor or the Fc portion of the antibody specifically, that he can target Treg cell without affecting effector T cells. And I think this approach is not only novel, obviously leading to top publications, but I think also make him pretty rich, right? So he started a new, I think he patented and started a company because of it. But I think that just let us realize, and we should all appreciate, the things that we study, and of course for a lot of good reasons, but also the things that it looked like periphery at the time, but they are actually also very, very important. And I don't want to spend another 10 minutes to talk about what other his contribution in terms of tumor immunology or immuno-oncology, because he has done a lot beside regular T cell work, including tumor neoantigen, a lot of other things. But I will spend too much time on it. So I think it's time to give Sergio to share his story with us. And I'm sure it's going to be a great talk. I look forward to it. Sergio, it's your time.
Thank you so much, Lifan. You almost made me cry. I think when I die, the scientific eulogy, it's all yours, man. That's the most impressive introduction I ever had. Yeah, so thanks very much for the invitation to me being a speaker in your department. It's an absolute honor. So I'm really grateful for the invitation. And I hope this darn internet makes it honor, because it's been good all day and now it's just floppy. All I'm gonna do though is to make one comment, because you're absolutely right, Lifan. I think that the whole talk goes about the premise of, we tend to go into topics. It's very common, right? And as students, we jump into the topic and we try to learn as much as possible, but it's usually hard to keep up. And we tend to forget that 10 or 20 years ago, somebody had done something similar or something that could influence what we're doing today. And for me, just so you guys remember, when towards the end of my talk, is that when we convinced Randy, that our old boss, that we needed to do T-Reg work in the lab, he said, okay, fine, deplete Tregs. I said, okay, so how do you deplete Tregs? Well, use an antibody. And in that time, the antibody was an anti-CD25. So I went into the freezer and I found the clone 7D4, right? And it binds. And I said, should it work? And he said, yeah, of course it's going to work. It's an anti-CD25. Just go and try it, like the classical, go and try it. And it never worked. So we almost dropped the project, right? 7D4, it's a mouse IgG, it's a rat IgM or a mouse IgM. And IgMs are don't deplete, right? But as a first year PhD student, not even paying attention to that, you just go and you do it. And then, yeah, of course, you go and you use the next antibody, which is a rat IgG2A. And sorry, I'm getting too technical, but this is important, which did deplete, right? So we took that rat IgG2A, which is a PC61, the one that probably all of you have used if you ever tried to deplete a Treg, and that it's a rat IgG1, I'm sorry. And that antibody depleted really well. We took mice, we injected with the anti-CD25, and the Tregs were gone in spleen and lymph node, right? And that was kind of, for me, the beginning of a path. And we'll touch upon those two facts, the 7D4 and the PC61, as I move along. Please feel free to interrupt me as I go. I think there's a lot of interesting concepts that could come out of this that I would prefer to share them instead of just to share the most important bits, instead of just being focused on in going through every single bit. Can you see that, but not as full screen yet, right?
Yeah.
And that will be the full screen?
Yeah, great.
Very good. All right. So I am an absolute believer that, although I'm not rich yet, Lifan, that basic science, basic understanding of what we do can lead to true therapeutic impact. And I think it's completely doable. And to me, my mission is to take any PhD student in the lab that is very interested in basic, foster that, but always give them the view that we can make a clinical impact. Obviously, you need hard work and quite a bit of luck, but I think it's doable. So just to set up the stage, when I started working in Jim's lab, what we were studying, it was the impact of anti-CTLA-4 antibodies, because that's the concept that he had developed. And looking at an untreated mouse melanoma model, which is a poorly infiltrated mouse tumor model, this was the classical picture. It's a tumor that had very few CD8s in red and had patches of infiltration that were primarily dominated by CD4 positive, FOXP3 positive cells, right? And being Jim's lab, needed to treat it with anti-CTLA-4 antibody. And when I joined, we moved away from looking in lymph node spleen to move into the tumor. And this was one of the first pieces of data that I generated in that lab, basically showing that when you give the antibody, in this case in combination with a tumor cell-based vaccine, you have a huge influx of CD8 cells in red. And when you quantify by flow cytometry, it looked like the Tregs were dropping. And we published a paper talking about the balance of effector and regulatory T cells and how important was that balance to understand whether a tumor was going to respond or was responding to anti-CTLA-4 therapy. That change in the balance. So here you have, I don't think, can you see my mouse? Probably not. Can you see it now? As a laser pointer? Yeah.
That's good.
Yeah. So again, some more quantification by flow cytometry. Untreated animals looking in the tumor have a low ratio of CD8s to regulatory cells. And when you give them the anti-CTLA-4 with the GVAX, with this combination therapy, you have a switch on that balance of effectors and regulators, right? And that switch correlated with about 70% of the mice rejecting their tumors. Because the concept and the dogma at the time was that CTLA-4 blocks inhibitory signals, either via competition or by blocking an inhibitory signal, that's what we thought that the antibody was doing, is that the ratio was being altered because you were accumulating effector T cells, right? So that was the predominant view in the lab for a long time. But the more and more we digged into the mechanisms, there was something that we couldn't understand. And that something was the fact that this was a very fast process. So this is a mouse, or two different groups of mice. One treated just with the vaccine. The vaccine doesn't induce an anti-tumor response, a potent anti-tumor response. And looking inside the tumor. And what you see here are the tumor infiltrating regulatory T cells. This is a TCR transgenic model. So we're tracking tumor reactive Tregs and tumor reactive effector cells. So this is a mouse treated with GVAX. A mouse treated with anti-CTLA-4 and then killed 12 hours later, it looked like all of the tumor reactive Tregs disappear, right? So it was really, really fast. And this was hard to explain by the hypothesis of there is so much increase in the CD8s that that's just giving you a change in proportions. This was suggesting, completely contrary to the dogma in the lab, that the antibody was having an effect classical or ADCC or ADCP. Right, so that was quite argued. And then I move into my lab at UCL. We're not sure about this, but we continue working together with Jim's lab and with my new lab. And at the end of the day, we decided to do a very simple experiment for those that are more familiar with antibodies. If you want to know if an antibody has ADCC activity, you do the experiment in mice that cannot do that activity, and the classical animal will be Fc common gamma knockout mice. So these mice cannot signal through the Fc receptor. And when you do that experiment in those animals, you treat with the anti-CTLA-4 and you don't lose the Tregs. So in one simple experiment, we demonstrated in mouse models that you require engagement with Fc receptors to eliminate the regulatory T cells in the tumor. So this was not only an alteration of on balances because the T effectors are expanding. This was a direct effect of the antibody by killing the Tregs through engagement with Fc gamma receptors. But we also demonstrated stories that this was NK independent, which means you could kill all the NKs and still get the activity. The data suggested, although this was complement independent, and that you require Fc gamma receptors, particularly Fc gamma receptor 4, which in mice is one of the most active Fc receptors, is usually found in tissues. What's important to mention also is that this antibody is a hamster antibody, right? It's a hamster anti-mouse CTLA-4 antibody, right? So you guys are going to ask me, well, what are the rules of engagement of a hamster IG to a mouse Fc receptor? Well, that's where the problem starts, right? We just take antibodies against a target and we validate a certain number of things, but at the end of the day, or at least 10 years ago, what we will do is we will move forward the antibody that give us the activity that we wanted, and the other ones we drop. So there is a piece there very important to understand. I think everybody has moved into that direction to really try to understand what the antibody can be doing. And a rat antibody into a mouse, a hamster antibody into a mouse, or a human antibody into a mouse, even against the same target, will have different activity depending on the isotype. And we used not to pay too much attention to that. So this is going to be very important. Now, the question there at the moment that we showed that the antibody could kill Tregs was, well, why is it killing Tregs? This antibody was designed against CTLA-4, which is cytotoxic T lymphocyte antigen number 4, highly expressed on CD8 cells. And that is absolutely true when you activate CD8 cells in vitro. But when you look at the expression of CTLA-4 on tumor-infiltrating lymphocytes, you get a very different picture. So this is looking at mouse melanoma tails. And in blue is the expression of CTLA-4, surface CTLA-4, on regulatory T cells. And in red, solid red, is the expression of surface CTLA-4 on the helper compartment, on the CD4 positive, FOXP3 negative. And I don't have it on this one, but if I was looking at surface CTLA-4 expression on the CD8, it would be equivalent to the dotted red line. So not surprisingly, in vivo distribution of the expression of these receptors is completely different from the in vitro distribution. So that's also something very important because we're trying to treat an animal and not a tissue culture dish, right? So that concept is also key. So what we hypothesize is that the antibody is not an antibody that specifically kills Tregs. It's an antibody like any other antibody ADCC enabled that will kill a target that expresses the most amount of molecules on the surface of that target. And that happens to be CTLA-4 on the Tregs. And the final demonstration on that story is that if we do, so we demonstrate that this was all Fc gamma receptor 4, the depletion of the Tregs. And this slide, this figure just shows you how important it is to the final anti-tumor activity of the antibody, because wild-type animals, you see here about 70% protection. But if you do the experiment in mice that are Fc gamma receptor 4, in which the antibody cannot kill the Tregs, and you cannot alter the ratio of effectors to regulators, you completely lose the activity. right? The dotted blue line. So there is no tumor protection. So the model we put forward is something along the lines of this slide. Regulatory T cells in the tumor express a lot of CTLA-4 on the surface, or more CTLA-4 on the surface than the effector cells. An antibody that is ADCC enabled will engage, or the cell that has the most amount of molecules, and that will allow trimerization of the Fc receptors, activation of ADCC or ADCP, and then killing of the Tregs. On the effector cells, that antibody is not going to be able to induce ADCC, or not efficiently induce ADCC. But remember, the antibody still blocks CTLA-4 interaction with B7. So we do think that the antibody still has that checkpoint activity, and those two things together give the maximal anti-tumor activity. But in mouse models, I assure you that to all of the antibodies described so far, if they cannot deplete the Treg, you're not going to get anti-tumor immunity. So Treg depletion is key to the function of these antibodies in mice in the context of mouse FCRs. So then Fred, a super talented postdoc in my lab, decided to push a little bit the envelope, because we were with all due merit, our findings were short of something. We're looking now at the biology of a hamster antibody in mouse Fc receptors, trying to make conclusions of human antibodies in the context of human Fc receptors. So we started our collaboration with Jeff Ravitch in New York, and Jeff had generated these mice, and instead of expressing mouse Fc receptor, they expressed the whole gamut of human FCRs, right? So that was half of our model. Fred is an excellent molecular biologist, so he took the variable region of the anti-mouse CTLA-4 antibody, the hamster, and he grafted it into different human IgG fraction portions, right? So he could generate an anti-human CTLA-4 that was very similar to that one in the clinic, an IgG1, which should be depleting, one with enhanced depletion, one without depletion. So we could ask the same questions, but in the context of human Fc receptors and human IgGs. That's basically what he generated. So sorry, just move something out of my screen. No, I can't move that. So basically, he generated a human IgG1 with a particular mutation that makes that antibody a pure checkpoint inhibitor, because that mutation will not allow binding to Fc receptors. So the antibody will only block CTLA-4. Then he made a variant, again, the same epitope recognizing portion, but the Fc portion will be a human IgG1, which is equivalent to the human antibody in the clinic approved by the FDA. And then he made a variant called the SDALI, which is one that has a set of mutations that enhances binding to Fc receptors, in a way to try to demonstrate the hypothesis that, well, if depletion is important, more depletion might be even better, or might create a better therapeutic. Long story short, he does all the experiments, the functional experiments, but they all match the tumor protection, and this is the tumor protection data. So mice that are left untreated, you have all of the tumors grow. Mice treated with an anti-CTLA-4 with a human isotype that is unable to kill Tregs. So this is a pure checkpoint inhibitors. There is a slight delay in tumor growth in some of the mice, but basically all of the mice need to be sacrificed. So just the blockade of CTLA-4 is not sufficient to drive anti-tumor immunity, or sorry, tumor rejection. And when he generates the checkpoint inhibitor, the depleter, which is a human IgG1, now he sees about 75–80% complete responses with 3 shots of the antibody. So the antibody requires binding to human Fc receptors in order to deplete and protect. And most importantly, when he developed in these mouse models an antibody with enhanced binding to Fc receptors, he gets an even better anti-tumor response. So again, mouse and human, mouse mechanisms and human mechanisms, the data does support the fact that Fc receptors are quite important for the activity of these antibodies. And we did a lot of experiments trying to demonstrate the expression levels of CTLA-4 in mice and humans. And as I told you, in mouse, it's preferentially on the Tregs. In humans, it's also preferentially on the Tregs, except for one little detail, which comes here. So we found out a piece of evidence that got us a little worried. So all of our data was pointing out to the fact that you should be making antibodies with enhanced ADCC to CTLA-4, because you will be depleting preferentially your Tregs. And that still stands absolutely, in my mind, it stands absolutely true. But it's the context, the bit that needs to be carefully considered. So this is looking in red at the expression profile of CTLA-4 and ICOS on human melanoma infiltrating lymphocyte. So here we're looking at human melanoma. And what you see here is that there is very high levels of expression of both co-expression of CTLA4 and ICOS on the tumor infiltrating Tregs, again, human melanoma. Those Tregs are likely very activated because are expressing lots of the CD25 receptor, of CTLA4 and ICOS. They also express CD25 and other co-stimulatory receptors. The helper, and that's the vast majority of the Tregs. The helper compartment, there is a fraction of them that do express CTLA4 and ICOS. And then the CD8 compartment, they express much less, but they do express some. So what we think is it's happening today is that the current ipilimumab antibody in the clinic has not been enhanced for ADCC. So it's a regular ADCC driver antibody, it's an IgG1. And that was not done intentionally. Don't ask me why, but that was the antibody, the isotype that was chosen for this drug. This drug was not designed to kill Tregs. People did not care about Tregs when this drug was designed, right? It was designed to block. So this isotype was the one that happened to give the best blockade in vitro. And that's the one that was moved clinically. But it is a human IgG1. Human IgG1s do interact with human Fc receptors, and they can mediate depletion. So we think that this antibody, it's targeting the Tregs, a fraction of them, targeting a fraction of that CD4 positive FOXP3 negative, which we think might not be so relevant because, again, this antibody works in the clinic, and sparing the activated CD8. But again, this is an hypothesis. Now, the issue or the concern that we had is that if you increase ADCC in this antibody, what you do, you move the threshold down. So you're going to have much better killing of the Tregs, which is what everybody will want, but you will start now killing some of your activated CD8s. So that made us quite nervous in the lab. Obviously, we're not biotech or not a company, so we presented this data, published this data, and move on into finding an antibody or a target that will be more specific and less controversial. Because this was quite tricky to publish, quite tricky to convince people that we were on the right track, that Fc receptors are important for the activity of these antibodies. So if we wanted to move into something less controversial, we did a terrible job because we moved to CD25, which is the high affinity IL-2 receptor. This was very much in vogue when Lifan and I were doing our PhDs, and then it was kind of eliminated and forgotten as a good target for Treg depletion. I'll explain why. So there are some new biological and mechanistic insights that were generated here. So for you guys, not much of an intro needed, right? 25 is a high affinity subunit. The whole receptor has also the beta and the gamma, and that's what signals into T cells, NK cells, and regulatory T cells as well. The antibody was really hot at the time because it's expressed at high levels on regulatory T cells and at low levels, CD25, on the effector and the helper compartment. But the moment that you activate T effectors in vitro, right, lots of data, or to viruses in vivo, the effector cells will upregulate, CD8 cells will upregulate a lot of CD25. So that makes you worry, exactly as I show for CTLA4, that you will not want to do that because your antibody might end up killing activated effector cells. Right. However, as I said before, it's very important that you check this in vivo, right? What is the distribution of CD25 in vivo? And this is 3 different mouse models of cancer. And what we're looking here is from poorly immunogenic tumor in the left to very immunogenic tumors on the right. So these are mutation-driven, carcinogen-driven mouse tumor models. And what you see, which is the MCA205, what you see here, very similar in all models, but in this one, to be more clear, is that the regulatory T cells, FOXP3, CD25, those guys are expressing really high amount of CD25 on their surface. Meanwhile, the CD8 compartment, there is a tiny fraction of them that express CD25, but much lower number of molecules, lower MFI. And then out of the CD4 positive, FOXP3 negative, there is a population that does express it. This is in tumor infiltrating lymphocytes again. So in my book, this delta between these guys and these guys, or these guys and those guys, it's a good delta to create an antibody that will preferentially deplete Tregs and not effector cells. We didn't want to run into the problem that we have with CTLA4, so we immediately went to look at the expression of CD25 in human TILs. And this is the expression of it. So this is patient with melanoma, non-small cell lung cancer, and renal cell carcinoma, treatment naive. In all of those patients, consistently, you see that CD25 in TILs, it's preferentially expressed on the FOXP3 positive population, and the CD8 cytotoxic population doesn't express much CD25. Right, so again, a big delta. And when we look at this, this is percentage of cells, but if we were to be looking at MFI, the distribution is very similar. So very few molecules of CD25 on the surface of CD8 cells. Now, the argument here is that we're looking at treatment naive. What will happen if these patients were undergoing immunotherapy? We were lucky enough that we could look at this. So this is a collaboration with the Royal Marsden. We have two clinical trials running, one in renal cell and one in melanoma, but very similar in terms of endpoint and in terms of data. I'm just showing you the renal cell carcinoma data. These are patients with kidney cancer for which we get a biopsy at baseline out of their tumor. The patients go on anti-PD-1, and then as they're being treated and responding or not responding to anti-PD-1, we get a second biopsy from the same site. So it's as close as you can get to a mouse experiment where you get a biopsy, a treatment, and then a second biopsy. And what you see here is the distribution of CD25 in brown, AIDS in red, and FOXP3 in blue. And as I hope you can see here, CD25 is preferentially associated to the FOXP3 signal, and the CD8 cells don't express much CD25. This is a baseline, and if the patient has been treated for 6 weeks with anti-PD-1, that distribution does not change. And when this is looked independently and blind by our histopathologist, Teresa Marafiotti, you see that both in the renal cell samples and in the melanoma treated patients, we see the same thing, that the CD8 compartment, either a baseline or on therapy, doesn't express much CD25, and it tends to be the FOXP3 compartment, the one that expresses it. So quite an important finding that either in treatment naive and in the context of immunotherapy, 25 is restricted or preferentially expressed on the regulatory cell and not the effector. This doesn't preclude that there will be some CD25 on the effectors. We cannot completely take out that hypothesis, but it's not detectable by immunohistochemistry, and by flow cytometry, the levels are extremely low. So a good target in my book, at least. So then why this wonderful target, why would this wonderful molecule was dropped as a potential target? And it was because of this type of data. This is just an example that we published a long time ago, but there are plenty of data like this. So when PC61, the mouse anti-CD25 antibody, the anti-mouse CD25 antibody, that happens to be a rat IgG1, was used, as I said before, we used it a lot. You take a mouse, you inject it with 400 micrograms of PC61. 4 days later, you look in lymph node and spleen, and the Tregs are gone. And there is a paper that showed that that is Fc gamma receptor 3 dependent. So the antibody is a bona fide depleter. So then what was the experiment that we all did? We took our favorite therapy. Ours in Jim's lab was GVAX and anti-CTLA4. And usually that therapy works really well at days 3, 6, and 9, but you delay it. So it's suboptimal, it doesn't work. And that's the black line, right? So that's our favorite therapy. And then you used to go prophylactically with the anti-CD25. You kill the Tregs, you verify at day 0 that the Tregs are gone, and then you give your suboptimal therapy, and you get this wonderful synergy. You say, you see, depleting Tregs is great, but then you do the real experiment, well, not real, but the less far away from the clinic experiment, and instead of depleting prophylactically, you challenge the mice with a tumor, you wait only 4 days, right? So question marks about how therapeutic this is, but it's after the tumor challenge. And then you deplete the Tregs and you give your suboptimal vaccine. And when we did that and everybody else did that, you get no synergy. So at this point, the field stops and says, the vast majority, that most likely the antibody is killing activated effector cells in the tumor, because they should, according to the book and everything we know about it, they should be depleting activated effector cells that upregulate the receptor. But I show you that in mouse, we don't see much CD25 on the effector cells. So there was a conclusion there, but without much data to support them. So Fred, again, in the lab, went and did the experiment. Is the antibody killing the activated effector cells? So these are untreated animals looking in the tumor, FOXP3 versus CD25. You see that in total, about 44% of the CD4s are Tregs. If you treat that mouse with the anti-CD25, yes, the CD25 Tregs are gone. This is not blockade. We're using another antibody clone to detect, but their Tregs are not gone. There is still 44% or near 44% of FOXP3 positives in that tumor. So the antibody is not killing activated effector cells. The antibody is failing to kill the Tregs, and what it was inducing is actually the downregulation of CD25, giving you a false impression that the Tregs were being depleted. So there was something wrong with this antibody that took us a while to figure out, that it wasn't depleting. But as I said, it was depleting Tregs in lymph node and spleen. that has been proven. And we looked at it and it was true, but not in the tumor. So at the same time, we were working on CTLA4. So we learned quite a bit about FC receptors. And we did a little bit more of analysis of the rat IgG1 anti-CD25 that we're using. And look at this. The binding affinity of a classical rat IgG1 to Fc gamma receptor 4, which is the important depleter in tissues, it's a no binder. So that rat isotype just doesn't bind to that Fc receptor, which is so important for depletion in tissue. Yes, the antibody binds to Fc gamma receptor 3, which is functions quite well in the periphery. And the other thing that we saw is that the antibody binds with very high affinity to the inhibitory Fc gamma receptor CD32B. So this suggested to us that the antibody was not optimal and that it could be optimized, it could be improved by simply the same way Fred did it for the human Fc receptors, to convert that isotype, that P61 rat IgG1, into a mouse IgG2A. And when he did that, he recovered binding affinity to all activating Fc receptors. He reduced the binding affinity to the inhibitory Fc receptor. So now he created an antibody that, according to the rules of the book, should be depleting. And that's what he tested on this experiment. So the mice get challenged with tumor, and then they get treated with the antibody, and then we check for Treg depletion. And here you see, again, untreated animals, lots of Tregs, treated with the rat IgG1, lots of Tregs, but down regulation of CD25. When you give the new version of PC61, the mouse IgG2A, now this antibody can properly engage with Fc receptors, can deplete the Tregs without a problem. So he fixed that issue, the issue of depletion. And again, we had completely ignored this because when this antibody was tested the first time, it was tested for the purpose of depleting in lymph node and spleen, and it does deplete in lymph node and spleen, but not in the tumor, because we didn't understand those rules of engagements between the rat IgG and the mouse Fc receptors. Now, will this fixing of the isotype lead to anti-tumor activity? And the answer is yes. So these are mice left untreated. This is a therapeutic intervention. So the mice have a tumor now, and then we treat with a single dose of the antibody when the tumors are palpable. And here we're combining with anti-PD-1 because we didn't think that it would work as a monotherapy. So these are anti-CD25 treated with the old isotype that doesn't deplete in the tumor. You get tumor growth. These mice are resistant to anti-PD-1, so you get tumor growth. And when you combine those two drugs, you don't see any synergy. Again, resistant to anti-PD-1 and the anti-CD25 cannot deplete Tregs. But when you give the new antibody that Fred made, the one that can properly engage with Fc receptors, remember, this is a single dose monotherapy, you see already two complete responses. And one of them grew a very large tumor and then it collapsed. Some other two mice grow very, very large tumors and then they collapse and then they relapse. So something was going on in these tumors. Not enough to say, okay, this is really impressive. But impressive was the fact that when we combine now anti-PD-1, which before gave us nothing, with one dose of the anti-CD25, we had this pretty nice, significant responses. Okay, so we could get proper Treg killing by solving the Fc receptor engagement and proper synergy now with this antibody. So that's the part that Lifan was making allusion at. The point we talked to Cancer Research UK. Well, I talked to them. I tried to convince them that we needed to file and protect the intellectual property on these findings, et cetera, and spend a year discussing with them. And the answer was always that you cannot file IP on mechanisms. You need to file IP on novelty. And there were plenty of antibodies already in the clinic against CD25 to deplete Tregs, but they didn't work. And they didn't work because they didn't get the mechanism. At least that was our view. Anyhow, after a long time of working with TRUK, we managed to engage with a small biotech, and we convinced them that this was a good concept and that we should take forward. So lots of paperwork later, they licensed it from us and we put the condition that we wanted to continue working on the basic science and the improvement of the concept. And the first meeting that we have with them after working on this thing for years and years, right, and getting this pretty data that I think is pretty, that I just show you, we have the first meeting and they say, is this the best you can do? You think you can make a better drug, right? After all this work, it was like, oh my God, you guys must be crazy. And that question actually was a very important one because we started looking at what was out there in terms of competition. So here we pivot a little bit more about thinking, okay, how are we going to compete with others? How are we going to have a proper impact? And what we realized is that the two antibodies that were in the clinic already, teclistamab and basiliximab, they are anti-CD25 antibodies. And they have failed to produce significant or striking responses in the clinic against solid tumors. Right? So that again is one of the reasons why people were not interested in CD25, always arguing that they were depleting the activated effector cells. When you look at those antibodies, they were not developed to treat solid malignancies. They were developed to treat graft versus host disease or autoimmunity or prevent transplant rejection. And, and to do that, they developed those antibodies to block the binding of IL-2 to the IL-2 receptor, hence the signaling into activated effector cells. And the only scenario that they've been used successfully against tumors, it was against T-cell lymphomas, because T-cell lymphomas, some of them depend on IL-2 signaling, right? So the antibody targets CD25, but was designed with a different intention, block IL-2 signaling. And IL-2 signaling, it's essential for effector T-cell function, right? So then that was kind of a moment that, oh, could that be? So then we look at our PC61 data and we realize that the P61 antibody, although we had fixed the isotype, also blocks IL-2 signaling. It's a weaker blocker, but it also blocks IL-2 signaling. So we thought, and this was an hypothesis, that perhaps we could improve this antibody, that perhaps something that is happening is that the antibody is targeting the Tregs, they express a lot, so similar to the anti-CTLA-4, those cells would be eliminated. The effector cells, the CD8s and some of the CD4s, we know they express CD25, but at very, very low levels. So we do think that the antibody will bind to them, but we have shown that it doesn't kill them. And that's probably because the density is low. But one of the things that happens when you kill Tregs is that you get excess IL-2 on site in situ. So if the antibody is blocking IL-2 binding to the IL-2 receptor, well, it might not be killing the effector cell, but we're wasting that extra IL-2. So that was our hypothesis, right? And daclizumab will be doing it even more. So what we propose to the biotech company and to CRUK is to use some of our funds to develop a new antibody that will bind to a different epitope on CD25, so we'll still kill the Tregs, but the different epitope will not block IL-2 signaling, right? And by not blocking IL-2 signaling, now we can have a loop of activation of the effector compartment. Again, an hypothesis. So we went back to the books, and lo and behold, we realized that the clone 74, which is the first one that I mentioned to you, the one that never worked at depleting Tregs, will never work at depleting Tregs because it's an IgM, but it was a non-IL-2 blocking antibody. So we took that antibody and we converted into a mouse IgG2A, so a classical depleter, and made, for the first time, an anti-mouse CD25 that could deplete potentially and would not interfere with IL-2 signaling. And this is some of the data on the IL-2 signaling. We call that antibody anti-CD25 NIB or NIB, non-IL-2 blocking. And this is a pSTAT5 assay done. You take T cells from a mouse, you spike them with a tiny amount of IL-2, and then you measure pSTAT5. And what you see here is that if you don't add anything, you get about 50% of the cells signal. If you give our 74 clone, our anti-CD25 NIB, you get no change in the IL-2 signaling. If you give PC61, you block 50% of IL-2 signaling. And as a control, if you neutralize IL-2 in vitro, you completely lose the activity. And we see very similar data when we do this experiment ex vivo. So bottom line, if we treat the mice, if we left the mice untreated and we look at CD8 TILs, you see very nice pSTAT5 signaling. If you treated the animal with the Treg depleting antibody that blocks IL-2, you lose IL-2 signaling. And if you treat with the new antibody we made that depletes Tregs but doesn't block IL-2, you have the IL-2 signaling. So we made an anti-mouse antibody that should be doing what we wanted to do. And the key piece of the experiment here is obviously to demonstrate that that antibody can induce anti-tumor activity. And this is the data. So these are a mouse model of colorectal cancer injected subcutaneously. If you left the mouse untreated, they all grow tumor. If you treat the mice with a single shot of PC61 mouse IgG2A, you get 10% complete responses, 10% delay, and then the rest of the mice grow. Remember, this is the antibody that if I combine it with PD-1, it works very well. But as a monotherapy, it doesn't work as well. And we think that that's, again, it's because the antibody, it's blocking some of the IL-2 that is being released when you kill the Tregs. So what happens when you give a single shot monotherapy of our new antibody? Well, in this model, and in every model that has a high effector T-cell infiltration, we get circa 100% complete responses. So single shot monotherapy can really, really well, deplete the Tregs the same way that P61 would do it, but give you a much more potent response. So we were all super happy with this. And obviously now we had a new composition of matter and new IP2 file that was quite unique to the field of Treg depleting antibodies. So this basically rescues the target as a potential IPable target. In terms of mechanism of action, we did a lot of work in this, but one of the things that we saw is that the CD8 compartment, and in some models, the CD4 compartment, when you give your single shot of the anti-CD25 nib, there is an increase in the amount of granzyme that the cells produce. And we just published a paper recently in Immunity showing that CD4 cells can acquire a granzyme or a cytotoxic program by when you alter the BLIMP BCL6 axis, and that's alterable by IL-2 availability. And in fact, if we combine the anti-CD25 nib with a neutralizing anti-IL-2 antibody, you completely lose the cytotoxic activity. So we do think that loop that we were proposing of extra available IL-2 is driving more cytotoxicity on the T cell compartment, and that's driving the more potent anti-tumor immunity. And the PC61 doesn't give you that as a monotherapy. So is IL-2 really doing the trick? So we did a couple extra experiments. These are blocking experiments to test the role of IL-2 in our system in the anti-tumor activity more than in just the granzyme activity. These are the mice on the control group, left untreated. These are the mice, a mouse group. It's about 10 mice per group treated with the anti-CD25 nib. You see very nice tumor control. And then we do anti-CD25 nib plus a neutralizing anti-IL-2, and you lose tumor control. Now, it is not surprising that IL-2 is essential, right? So it's kind of a funky control. It just shows that IL-2 is critical. But the key experiment was this one. The yellow line, it's the mix of the anti-CD25 nib, the new antibody, with PC61. So here we're getting very good Treg depletion across the board, but the P61 is doing that IL-2 blockade. And when you have that, you completely lose the activity of the antibody. So this demonstrates that the problem with PC61, and likely the same problem will have happened with daclizumab and basiliximab, is that they're blocking IL-2 signaling into the effector T cell. And then we run a set of many experiments just now to please ourselves, but also and equally important, pleasing the reviews, right? So how does this work across a range of tumor models and conditions, etc.? So one of the things we wanted to try is the activity of this antibody as a single dose or multiple dose against larger tumors. So here we're intervening when they're about 250 cubic centimeters, and you see those are control animals. All mice need to be sacrificed. This is a single dose of the anti-CD25 nib against these larger tumors. It's quite interesting. They continue growing, so this will be in humans pseudo progression. And we think that that's because it's altering the balance inside the tumor, and then you start seeing that very nice collapse, and then some mice, some tumors escaping. So we treated that with multiple doses weekly, and we see now circa 100% complete responses. And this was quite important because going into the clinic, it's going to be hard to justify a single dose of the antibody. I think pharma will want to try multiple doses. And the argument that was always, it's always the argument, well, your effector cells might have regulate CD25, so then with your second dose, you will kill them. And this actually shows that that's not the case, that you can give one dose a week of this mouse IgG2A, and you don't have a detrimental effect. You actually have better anti-tumor immunity. And we had a model for acquired resistant to immune checkpoint blockade, which is basically mice that are left with larger tumors, and then they're treated with anti-PD-L1. And what you see there is that, so if they're untreated, they'll grow tumors really quickly. If you treat them with anti-PD-L1, you get a slight delay, so about almost 10 days of flattening and stop tumor control, like they stop growing, but then they synchronize, and pretty much all the tumors just shut up, just like start growing like crazy. So all of the mice become resistant to anti-PD-1 post an initial set of responses. So we said, okay, let's intervene with the anti-CD25, one shot right at the middle of the anti-PD-L1 intervention. And when we do this, we move from a scenario that we have 0% complete responses to rescuing about 50% of the mice. So we do believe that this antibody would combine quite well with anti-PD-L1 to prevent acquisition of resistance. And as I mentioned, I think I mentioned before, this works pretty well for very infiltrated tumors where you have already a high ratio of effectors to regulators, but then you will want to have an impact in poorly infiltrated tumors. And the poorest method to get poorly infiltrated tumors a little bit more infiltrated is using vaccines. And in our lab, our favorite is um, legacy from the Allison lab, our lab uses GVAX or tumor cell-based vaccines as a cheap mean to test vaccine combinations. And that's what we did here. This is a mouse model. This is the B16 BL6 that we use classically with anti-CTLA4. This is very resistant. At the setting that we're giving it here, this model is resistant to GVAX and anti-CTLA4. And what you see here is that the GVAX alone on those mice don't do anything. The one shot of the anti-CD25 nib gives you a little bit of delay in tumor growth, but all of the mice needs to be sacrificed. And the combination therapy is quite interesting. And this interesting for us is the fact that we see now resistance, and resistance allows us to study mechanisms of resistance and potential combinations. So this is why I like this piece of data. We don't want to cure 100% of the tumors because we want to study resistance in the future. So this mouse gives us about pretty much 70% of the mice, they all undergo a first set of response. If I were to magnify in this area, you will see that the tumors grow to a large size and then they collapse. And then little by little, they start relapsing. So overall survival significantly increases, but there is a resistance there. But the point is that the combination of a vaccine and a single dose of anti-CD25 NIP can get you a good anti-tumor response. And what's really cool of this is that you can do unsupervised, very complex analysis of that tumor microenvironment by flow cytometry. And just some vignettes coming out of that analysis, looking at the overall cellular populations within the tumor and the changes that we observe when we compare untreated versus monotherapy CD25, or untreated versus monotherapy vaccine, or untreated versus the combination, are as follows: is that we see usually a decrease on, we see a decrease on the Tregs, but also on a population of CD4 cells that are PD-1 high. With the monotherapy anti-CD25, those CD4 PD-1 high have been described by Jet Wolchuk's lab as suppressive. So it's quite interesting. There are not FOXP3 positive cells, but they're deemed to be suppressive, and the monotherapy antibody takes those down. Meanwhile, it increases the frequency of activated effector cells in the tumor. The GVAX alone, what it drives primarily, it's infiltration by activated effector cells. So it obviously doesn't affect the Tregs. There is a slight increase in Tregs when you give GVAX, and that's why we think the combination is so good, because the combination keeps on driving the elimination of that PD-1 high helpers. It drives a lot of activated effector cells into the tumor, but it also drives, which we think is quite important, a subset of NK cells to accumulate in the tumor, activated NK. So this antibody is not only affecting adaptive immunity, we think that it will have an impact on innate immunity through recruitment of activated NK cells. And now we know also macrophages, although not in this publication. So vaccine plus anti-CD25 and IB drives Treg depletion, as well as the CD4 PD-1 high, effector cell infiltration and activation, and innate cell activation. So all of this led in pretty much in parallel, as we were developing the concept of the anti-CD25 NIB, we're working with this biotech developing the human counterpart. We said, the data is good, right? We have a good surrogate. Let's make the anti-human antibody. That was the intention of this collaboration from the very beginning. So we created an antibody. We didn't. We ordered generation of an antibody to a company called Adimab that using yeast display, they generated a very large number of binders. And then based on our preclinical data, we said, we want the following features. We want very good binding to anti-CD25. We don't want any blocking whatsoever of IO2 signaling. So we gave them our pSTAT5 assay. So they will use that as a screening method. And then last, we want the ones with the best ADCC activity. So it was absolutely rational design based on mouse data. So makes you worry, right? So if you don't believe that mouse data can inform anything, that's what we did. And when they generated all the candidates, we also decided at the end, and this was, I wouldn't say a guess, but it was a bet, and based on the data, that we wanted to have enhanced ADCC, right? Because we believed that the pattern was so clean on effectors versus regulators that we would preferentially be killing the regulators and not touch the effectors, even with the enhanced ADCC. And that's just the crystal structure of the antibody. This is the IL-2 receptor alpha. This is IL-2 in purple. This is the beta and the gamma subunit. This is looking from the top, and you see RG6292, which is the anti-human antibody, binds to the opposite phase of the CD25. And that's why it doesn't intervene with the binding of the receptor to IL-2 and signaling. At this time, as we were generating all this data, the little company was acquired by Roche, which made us very happy because now we had somebody to support the toxicity and clinical development of the antibody, right? So now we were in serious business, and we continue working with Roche on the mechanism of action of the human molecule. And we run a battery of tests very similar to what we saw in mice. How am I doing with time, Lifan? I have only a couple more slides.
You still have 6 minutes, but we want to leave some minutes for people to ask questions.
Okay, so just capping up on this, we said we wanted RG62 to be non-blocking of pSTAT5. This is PBMCs on our pSTAT5 assay, so we drizzle a little bit of IL-2, and you see here daclizumab as a control completely ablates pSTAT5 on all T cell compartments. Meanwhile, RG62 doesn't affect pSTAT5. When we put it into a killing assay in vitro with PBMC in a dose-dependent manner, our anti-human CD25 kills the Tregs, and only a very, very high amount to start killing the activated effector CD4s and CD8s. We took primary single-cell suspensions from human lung cancer and human colorectal carcinoma samples, and we tested the ability of antibody to kill Tregs. And there you see the antibody kills very well. And if the antibody is Fc silent, it cannot kill. And in this tumor type, it also kills very well. And if it is Fc silent, it cannot kill. And then we use mouse model, a humanized mouse model of cancer, which I know has several caveats, but we wanted to push a little bit further into the type of models. And in this is, again, it's CD34 progenitor reconstituted NSG mouse, right? So we'll reconstitute some human T cells and myeloid cells. And this mouse is challenged with a prostate cancer cell line. And after some time, these mouse are left untreated, and then we look at the tumor microenvironment. This is CD25 versus FOXP3 on the vehicle. This is on the CD8 compartment. We treat the mice with our anti-human CD25 antibody, and you see very nice killing of the CD25 FOXP3 positive cells, some FOX left. You see in terms of PD-1 and CTLA-4, the CD8 compartment now inside the tumor is quite activated, right? You depleted the Tregs, and the CD8s now have upregulated PD-1 and CTLA-4. And we put as a comparison EP, and EP, the anti-CTLA-4 antibody, killed in a less specific way the CD4 Tregs, but it killed them. But what's quite important and consistent with what I mentioned at the beginning, that population of activated PD-1 high, CTLA-4 high cells are gone, right? So again, concerns about an ADCC-enhanced anti-CTLA-4 antibody driving potential detrimental effect. And that's a quantification. So we believe that we created an antibody that, a concept that comes from mice that shows that CD25, it's a good target, but then you need to target it without blocking IL-2. And parallel to all the murine data, we created a human antibody that has the same features, exactly the same features, or was built at the image of the mouse surrogate. And that antibody entered a clinical trial, a phase 1 basket trial in November 2019. They just opened a second trial a month ago on combination with anti-PD-1 from Roche. So that means that the first trial probably is going well if they're opening a second one. And you will be probably hearing some data about those trials at the AACR meeting coming next month. So conclusions, we believe that CD25 is a selective target for therapeutic Treg depletion, even in the context of anti-PD-1 immunotherapy in humans. The failure of the previous anti-mouse CD25s was due to poor understanding of the role of Fc receptors and the fact that we use these mixes of hamster antibodies into mice or rat antibodies into mice without taking that into too much of a consideration. We can fix the old PC61 by converting it into mouse IgG2A, but that antibody still partially blocks IL-2. So to make the best surrogate, you want a good depleter that doesn't interfere with CD25 and with IL-2 signaling, and that's the antibody that we made in mice, and we made that same version also for humans. And last but not least, my team I think this was really a collaboration between a lot of people in my team, people in that tiny biotech called TAS Therapeutics, and then the big pharma company Roche. Isabel Solomon was a tech that became a master's student, that became a PhD, and she led this project throughout. So for a PhD student to have one of her chapters of her thesis being part of a clinical trial, I think that she was over the moon, and we're very proud of her and everybody else that contributed from Tusk, from Roche, the team at the Royal Marsden with the clinical trial and the clinical trial samples, and everybody else, including especially Cancer Research UK for the support, Tusk, and the grant from Roche as well. Thank you very much.
Thank you, Sergio. So we have some time for a few questions. Anyone have questions?
Question.
Oh, Eric.
Oh, hi. Thanks for so it's very interesting. One of the obvious question, I think the problem is that we know that Treg is very important for the our immune tolerance. So for this antibody, do you have any idea like whether you can like specific target Treg in the tumor only? Is it a possible way?
Yes, so that's a great question. So the answer, to give a short and answer more questions, there are more. I would love to target this antibody to the tumor. There are ways of doing that with antibody engineering. The poor man's method is to make bispecifics, and we made a couple, and they're being tested right now. The first one is an anti-PD-L1, anti-CD25. It's not going to restrict it to the tumor, but at least it's going to enrich it in the tumor. And there are several other means that we're testing, but there is lots of extra engineering to doing the antibody.
Great, thank you.
And I should add, so for the paper, in the paper, we included the toxicity data in cynos, in macaques, and there was no tox. There was no tox profile at very high levels of the antibody. So that was actually quite interesting.
Okay, got it. Thank you.
Sergio, I have a question myself. So you said that the reason it doesn't kill the effector cells is because probably because the density of the CD25 expression on the effector cell compartment. But when you show the Treg depletion data, you show a clear depletion regardless the level of CD25 expression on Tregs. Because if you really look at Treg, you still have CD25 high and CD25 intermediate. But if you go back to this, yeah, the humanized data. I mean, I think this is the most clear one. If you look at the middle part, right? Because if you look at the vehicle, you can see there's a CD25 intermediate FOXP3 positive population, just like the FOXP3 negative population. But if you look at the middle part, almost all FOXP3 positive CD25, regardless the level of expression, gone. While the CD25 positive FOXP3 negative population remain there, even though they have the same level of CD25.
So I would interpret this a little bit different. So if you, or at least this is how we interpret it, and it's a feature of the antibody, which I cannot disclose too much without getting in trouble, but it's obvious from this data. If you make a line here, right, you're taking down all of these guys and almost like a flat line, whatever few CD25 high FOXP3 negative cells are. And you're taking out all of your very CD25 high and FOXP3 high cells. So also connected to the previous question, we believe that due to the affinity of this antibody, which is not the highest in the world, this antibody takes preferentially the very high CD25 population. And that makes it even better when it comes to the tox profile. So I do think that there is, and we have data, and you can see it here, the CD25 is expressed on the helper and cytotoxic at lower levels, but those cells are not significantly being impacted by the antibody because of the affinity of the antibody.
Yeah, that I agree. But my question is the CD25 low, the intermediate FOXP3 positive population still get eliminated. Look at the FOXP3 positive side, not the FOXP3 negative.
This one. Yeah.
Like the majority of them also get eliminated if you compare to the vehicle control.
Yeah.
So that is the question I have is, it worked great, right? It target Treg specifically, but I just don't think it can be simply explained by the expression level of CD25 because for some reason, CD25 intermediate FOXP3 positive Treg cells still get eliminated better than the FOXP3 negative population.
It could. I think that that's a good point. This is a representative plot, and I don't want to poop over my own data, but I think it would be good to look at this type of plot for every single mouse we have done, to be more thorough. I don't know how representative this part, the fact that we're eliminating these guys here according to this flow plot, is how quantitative it is. But yeah, that's it. That's a good point. I would have said that for the old antibodies, the one that block IL-2, yes, you had that double ability of killing, the two mechanisms to kill Tregs. This one seems to be a bit cleaner. Now, it could be inducing downregulation of the receptor as well. We know that that still happens to a degree, and that forcing also some killing of those Tregs. So yes, there could be other mechanisms apart from just the density, the killing the CD25 high. Yeah, you're right.
Well, I mean, I don't know whether you want to look into this direction. And one of the possibility is the CD25 intermediate population. Once a high population get deleted, because the abundant IL-2, they go up,
yeah,
and then they get deleted.
Yeah,
right.
Yeah,
so
it could be. So what we're doing now on that B16 melanoma project with GVAX, we're tracking TCR repertoire longitudinally and also single cell RNA-seq to try to understand the dynamics of the different populations. So there, if we're lucky, we might be able to pick up the different, using T cell receptor as the barcode, the different populations of regulatory T cells are being eliminated, maybe the upregulation of CD25 as you're suggesting, and then potential elimination.
Okay. Do we have any other questions? Okay, I don't see anybody raise your hand. So if there's no other question, thanks Sergio again for the wonderful talk. Thank you very much. And I thank everybody for coming today. We have really a good attendance today. Okay, so
thank you very much, everyone.
You're going to meet with Ye Zhen, I believe, right? Based on your schedule. Okay, very good.
Yeah, but it's a different meeting room, right?
Yeah, it's a different meeting room. So that's just say goodbye for now.
Okay, so it's a different meeting room. That's all gonna be on my calendar, right?
Yeah, you should be on your calendar. You should have the link.
Okay.
Okay. Thank you.
Bye. See you guys in a minute. Bye-bye. Thank you, everyone.
Bye-bye.